Aloha and welcome to our video on acids, bases, and a little bit about the pH system. Today's video we are going to talk about the roles of acids and bases in chemistry and living things. We'll talk a little bit about the pH scale and where acids and bases fall on them, and then finally we'll talk a little bit about buffers. Okay, first up, let's talk about acids real quickly. Acids are substances that donate a hydrogen ion or they can accept an electron pair. Some common examples for us to think about are stomach acid. Some of the other ones you've probably heard of hydrochloric acid or sulfuric acid. Notice they have this extra H here, and that's kind of the clue that gives us the idea that it's an acid. Now, bases are about the exact opposite of acids. Acid could donate a hydrogen ion, a base will accept this hydrogen ion, or it can release this hydroxide ion. Okay, notice it has a negative charge. They also can donate an electron pair, and they're often referred to as alkali. Some common bases, the biggest one of all is sodium hydroxide, and you'll notice it has this OH, this hydroxide group in the name, and that kind of gives us a little clue that it's a base. Okay, so let's look at the differences between acids and bases. Remember that they are opposite of each other. So for an acid's going to accept an electron. A base, on the other hand, will donate an electron. An acid donates a proton. The base will accept that proton. Okay, acids generally tend to break apart into this hydrogen ion, while our bases form these hydroxide ions. And you can see some examples over here. We talked a little bit about lemon juice. Our stomach acids are filled with them. Our nucleic acids are acids down here. And then we talked a little bit about bases. We talked about sodium hydroxide. Then baking soda would be another one here. Okay, so if we want to see how strong or weak an acid or a base is, we use something called the pH scale. The pH scale runs from 0 to 14. And right here in the middle is where we have something that is neutral. Okay, and that's going to have a pH of 7. And the best example we have of neutrality is this one right here, this pure water. Now remember, if we look at the idea of water and its formula, it is a hydrogen, which we said makes an acid, and it is a hydroxide, which makes a base. So you add those together and we end up with something neutral, which kind of makes sense to us here. Okay, and as I alluded to, we have our acids. Our acids range from 0 up to 7. Remember, at 7 it's neutral, so it's not quite there. And some examples, like we said, stomach acid is a pretty strong acid, vinegar is one here, and even what comes out of our tap water tends to be slightly acidic. Now our bases are going to have a pH greater than 7. Okay, so these ones here are going to be greater than 7, and things like our soaps are going to be 1, and then lye is a real strong one here. We talked about sodium hydroxide. Okay, so our pH scale goes from 0 to 14. Those with numbers lower than 7 are going to be acids, and those with numbers greater than 7 are going to be bases. Okay, the last topic we're going to talk about today are going to be buffers. What a buffer does is it kind of restores neutrality to a weak acid or a weak base. And that's going to be really important if things like blood, where blood wants to be about neutral. So if it gets too acidic, we can die, and if it gets too basic, we can die. And it doesn't have to get very far. What a buffer does is we add it into our solution, and then what it'll do is it'll kind of break apart the acids and the bases, and they'll form a little bit more of these waters, and that just kind of dilutes it down and makes it more neutral. So key thing, we use a lot of buffers when we're talking about biology, and we'll go through those just a little bit, and they'll go through them a little bit more in the lesson. Okay, that takes us to the end of our video today. Hopefully you learned a lot. Good luck on the lessons, and we'll see you next time.